Dear students, in the last class, we have studied about the nuclear fission. But today, let us study about the nuclear fusion and uses of radioisotopes. The process in which the two light nuclei combine to form a heavy and most stable nucleus with the liberation of an enormous amount of energy is called nuclear fusion. So in nuclear fission, heavy nucleus splits into the two lighter nuclei, but in nuclear fusion, the two lighter nuclei fuses to form the heavy or the bigger nucleus. Example, if I take the 4 protons, they combine and gives the helium nucleus with the liberation of a positron and the energy. So this can also take place by taking the deuterium, and then when it combines with the tritium, so we get the helium nucleus and also the neutron with the liberation of the large amount of energy. The energy liberated in nuclear fusion is much higher than the energy liberated in nuclear fission. During nuclear fusion, there is loss of mass which is converted into energy. Similarly, even in nuclear fission also, there is a loss of mass which is converted into energy. The mass defect in the case of nuclear fusion is far greater than in the case of nuclear fission. Hence, energy released in fusion reaction is far greater than the fission reaction. Nuclear fusion is also called thermonuclear reaction because it takes place at very high temperature, that is around 200 million Kelvin. Just you imagine what about the temperature. So the temperature that is present in the gas what we use in the house, it's around 400 to 450 degree. This much temperature is available in the core of Sun and other stars. Hence, nuclear fusion is possible only in Sun and other stars. The fusion reactions taking place in the Sun are, the 2 protons, they combine to form a deuterium and we get a positron with the energy. And another possible reaction is the deuterium combines with the proton and forms the helium nucleus with mass 3, He2 and gives energy. Here I have shown about the nuclear fusion. So in this, the nuclear fusion takes place in the star. So in this, the nuclear fusion I have shown below. So how the deuterium and tritium, they combine and forms helium with the liberation of the energy. So other possible reactions in the Sun, that is He2 combines with another proton to form He2 and the positron and the energy. The overall reaction is the 4 protons combine to form the the helium nucleus and 2 positron, and with the liberation of energy around 26.7 million electron volts. The highly destructive hydrogen bomb is based on nuclear fusion. Coming to the Uses of radioisotopes. Radioisotopes are produced in nuclear reactors. These isotopes have several applications in the fields such as agriculture, medicine, industry, chemical analysis, reaction mechanism, dating of fossils and rocks, and so on. Radioisotopes can be used as either a source of radiation for diagnostics as well as therapeutic purpose, or as a tracer element for the study of certain reaction mechanisms both in biology and chemistry. Now let us be restrict to the radioisotopes in tracer technique. Tracer technique is extremely useful in the study of reaction mechanisms, structure of compounds, nature of linkage, kinetics of chemical reactions, solubility of sparingly soluble salts, etc. The radioactive element used for this technique is called tracer element. Radioisotopes used in tracer studies are beta emitters. Generally, they are all the beta emitters. In the tracer technique, the substance to be investigated is labeled with a suitable isotope of an element of the molecule. This is introduced into the system under examination and experiments conducted as usual. The resulting products are then analyzed for the presence of the tracer isotope using Geiger-Müller counter. This technique was first introduced by G. Hansey and F. Penneth in 1913 to determine the solubility of sparingly soluble salt such as lead chromate. Tracer technique in chemical analysis. By tracer technique, it has been possible to establish that chemical equilibrium is a dynamic process. So in earlier class, students, you have studied about the chemical equilibrium, which is dynamic in nature. How to illustrate it? So for this, let us take an example, the lead iodide in solution. It is in equilibrium with lead ion and the iodide ions. Saturated solution of lead iodide in contact with solid lead iodide are prepared in separate beakers. In one of them, the solid lead iodide containing radioactive iodide, that is iodine 131, is mixed. After some time, the small amount of saturated solution of this beaker is transferred to the another beaker. Since both are saturated solutions, the concentration of the solid remains unaffected. After some time, the solid lead iodide, originally non-radioactive, becomes radioactive. This process shows that this reaction or this equilibrium is dynamic equilibrium, in which the radioactive iodide 131 ion in solution substitutes to non-radioactive iodide ions in the solid. Let us take how this tracer technique is used to other reaction mechanisms. Consider the hydrolysis of methyl acetate resulting in carboxylic acid and alcohol, which involves whether it is a breaking of the CO bond either by a process 1 or by a process 2. Now, for this, so let us, you see here, I have shown the methyl acetate CH3COO, CH3 which on hydrolysis gives CH3COOH plus methyl alcohol. Whether the bond breaks in first stage here, whether it breaks here or whether it breaks here. So this can be confirmed by this experiment. By using water enriched with oxygen 18 isotope in the reaction, after some time it was found that radioactive oxygen 18 was found only in acetic acid and not in methanol. This clearly indicates that the breaking up of the CO bond is as given in the first case. So this is how one can use the radioisotope tracer technique to identify the reaction mechanism. Coming to the radioisotopes in agriculture. Utilization of phosphates from soil has been studied by radioactive tracer method. Phosphate fertilizer is mixed with radioactive phosphorus 31 and studies on the extent to which the plant at what stage absorbs the phosphate more is carried out. It is found that phosphate absorption is more, around 60%, when added very early at the time of sowing than at a later stage when the absorption is only 35%. This shows that the phosphatic fertilizers should be added in the initial stage, not at the later stage. By using radioactive calcium 45 as a tracer, it has been shown that the absorption of calcium by the plants from the soil is same from calcium oxide as well as calcium carbonate in acidic medium. The absorption of calcium from calcium sulfate is much less unless the soil is markedly acidic. Yes. Here I have shown the applications of radioisotopes. So in the agricultures. So in the agriculture, the research tools to develop new strains of crops, and it is also used that track uptake of fertilizer from roots to leaves in a plant. So here I have shown this. So in this slide I have given about the agriculture, so applications of these radioisotopes. Nitrogen 15 or phosphorus 32 is added to soil water. After the plant absorbed the soil water, the radioisotopes can track uptake of fertilizer from root to leaves. Radioisotopes are used to kill pests and parasites and to control the ripening of even the fruits. So it also, the radioisotopes induces genetic mutation in plants and gives better strains, higher resistance against diseases. So this is the another slide. So the application of the radioisotopes. So a Geiger-Müller counter is used to detect the movement of the radioactive phosphorus 32 throughout the plant. This information helps scientists to understand the detailed mechanism of how plants utilizes phosphorus to grow and reproduce. So even in agriculture, we have various other applications. Radiation can be used to control insects in grains and fruits. So these inhibit the sprouting in tubers, delay the ripening of some fruits and vegetables. The radioactive isotopes are used. These are also used to reduce the problems of parasites in production of animal origin. It delays mold growth on strawberries and other fruits. Radioisotopes kill microorganisms and insects in spices. Commercially sterilized foods destroying all microorganisms of public health concern. That is, the special diets for people with weakened immunity system. So these things are very good for them. Now coming to the radioisotopes in medicine. In the study of obstructive vascular diseases, radioactive sodium chloride containing sodium 24 isotope is injected into the bloodstream as a salt solution. To move sodium chloride from the forearm to the foot, it takes half an hour for normal person. It takes longer time for the persons suffering from obstructive vascular diseases. To test the activity of thyroid gland, radioactive iodine 131 is used. In case of hyperthyroidism and cancer of thyroid gland, iodine 131 destroys abnormal tissues more rapidly than the normal tissues. A malfunctioning thyroid can be detected by giving a patient to drink a solution of sodium iodide containing a radioactive iodine of known activity and further measuring the activity just above the thyroid to see if iodine has been absorbed at the normal rate or not. The gamma rays coming from cobalt 60 isotope are used to cure cancer in radiation therapy. The gamma rays coming from the iodine 124 isotope is used to image the brain. In the treatment of blood disorders, radioactive phosphorus in the form of phosphate is used. Phosphorus 32 is also used in the treatment of bone diseases. Iron 59 radioisotope is used in the study of iron metabolism in the spleen. Potassium 42 radioisotope is used for the determination of exchangeable potassium in coronary blood flows. Coming to the radioisotopes in food preservation. Food irradiation is a method of treating food in order to make it safer to eat and have a longer shelf life. This makes food safer to eat and keep for longer period. The gamma rays coming from cobalt 60 isotope are used for irradiation. Irradiation is also used to delay the ripening of fruits and sprouting of vegetables. Dear students, remember the people going to the space months together and years together. So the irradiated the foods are taken there, and a chapati what you prepare in your house, you can store it months together, so if it is irradiated with gamma rays. Similarly, if you irradiate, the ripening of the fruits gets delayed, so you can keep the fruits months together. So this is the major application of the radioisotopes in food preservation. Coming to the lastly, another important one is the radioisotopes in carbon dating. Knowing this technique, we can find out the age of the fossil fuels. The carbon dating technique was developed by Willard Libby at the University of Chicago in 1949 and won the Nobel Prize in 1960. This technique is used to find out the age of archaeological objects or fossil fuels. All living matter contains a definite amount of radioactive carbon, that is C14 isotope, present in the atmosphere. It is formed in the atmosphere by the action of cosmic rays on nitrogen. When cosmic rays containing the neutrons, when it hits to the nitrogen, we get carbon 14 and a proton. This carbon 14 on oxidation forms radioactive carbon 14 dioxide, which mixes with the normal carbon dioxide, that is C12 carbon dioxide of the atmosphere. Since carbon 14 decays constantly, the ratio of carbon 14 is to carbon 12 remains constant. Plants absorb the C14 carbon dioxide and C12 carbon dioxide due to photosynthesis. As animals eat plants, this also enters into the animals. Hence, the ratio of the C14 carbon dioxide and C12 carbon dioxide in living plants and animals remains the same as in the atmosphere. When the tree is cut or animal dies, C14 is to C12 cycle stops, so that carbon 14 starts decaying to nitrogen 14 and a beta particle. The equation is shown there, and half-life period of this is 5770 years. The ratio of carbon 14 to carbon 12 decreases which becomes a clock to measure the time after the death of the plant or animal. Average carbon 14 content in a fresh piece of wood is found to be 15.3 plus or minus 0.1 disintegration per minute per gram. Samples having the age up to 20,000 years can be accurately calculated from this method. So this is about how to find out the age of the fossil fuels, the methodology. So these are the various applications of radioisotopes. Here in this slide also have given the applications of the various radioisotopes, like cobalt-60 is used in radiation therapy to prevent cancer. Iodine-131 is used to locate a brain tumor, monitor cardiac, liver and thyroid activity. Carbon-14 isotope is used to study metabolism changes for patients with a diabetic gut and anemia. Sodium-24 is used to study blood circulation. Thallium-201 radioisotope is used to determine damage in heart tissues, detection of tumors. So like this, so the various, the other things are also given. In this slide, isotopes used in medicines. Cobalt-60 is used for external beam radiotherapy, now used more for sterilizing purpose. Erbium-69 is used for relieving arthritic pain in synovial joints. So like this, the iodine 125 is used in cancer bronchotherapy and also so many other things. This is a table once again shows the various isotopes, their half-life periods and the uses where these isotopes are. Yes, this is one more slide of the medical radioisotopes. The isotopes used in the medicines like sodium-24 is used to study general biological processes, iron-59 diagnosis of blood diseases, technetium-99 diagnosis of various diseases, cobalt-60 treatment of cancer, the strontium-90 treatments of tumor, iodine-131 so treatment of thyroid cancer and so on. Yes, this is the one more slide which shows how this nuclear medicine clinical uses. So in this, the nuclear medicine clinical uses, so I have shown here the instrument. So this is called MRI mirror imaging. Nowadays it is very common. So here the person will be put here and he is allowed to enter into this. So in this case, this is a magnetic field. So that magnetic field generates and whole image of where he wants in which particular point. So it gives a 3D image. That's why it says it helps in early diagnosis, determines severity of diseases, it helps to select the most effective therapy, and it determines patient's response to therapy and identifies reoccurrence of diseases. So like this MRI is very useful technique. So here, this is a slide where I have shown the nuclear diagnostic applications and also the various other radioisotopes used for controlling the hormones systems in the body. So the body produces so many hormones. So all these hormone regulations can be controlled with the help of the radiotherapy. So here, this is a slide I have shown the radioisotopes in food preservation. So I have shown here so many the vegetables. All these vegetables you can keep it for the days together. So just by irradiation with the cobalt-60 isotopes of the gamma rays. So what you eat, the pizza and various other the foods, just you irradiate, these can be stored for months together. That's why in India the traditional people, they say don't eat such things, eat the fresh ones, because these things have been made long back and it stores. And of course it is hygienic only, but it is not fresh. So this is the concept what the Indian people, so they don't like these aspects. Of course, today everyone uses this. So here I have shown the slide using the gamma rays. So you can store these the fruits for longer period, and how this irradiation is done, here it is shown here in the above slide. Yes, here in this slide I have shown what is this food irradiation. Food irradiation is a process in which food products are exposed to controlled amount of radiant energy to increase the safety of the food and to extend shelf life of the food. Like pasteurization of milk and pressure cooking of canned foods, treating food with ionizing radiation can kill bacteria and parasites that would otherwise cause foodborne diseases. So this is one more slide, that is food irradiation and their uses where these things are used. So it is reduced insect infestations in the grains, spice, fruits and vegetables. It inhibits sprouting. It retards ripening, inactive parasites, eliminates spoilage microbes, and so on. So this is the applications of this food irradiation. So in the carbon dating, what I have said, so this I have given in this diagram. So here this, what is this carbon dating means? So the sun, so with the help of a cosmic rays, the nitrogen 14 turns into the carbon 14. So this is how what it does. So through the sun, the cosmic rays, so they combine with the nitrogen and forms the carbon 14. This carbon 14 is taken by the plants and also the animals, and carbon 12 to carbon 14 ratio is fixed in them. When the plants and animals, they die, they will be buried or they put into the soil, and there carbon 14 is to carbon 12 ratio disturbs. Following death and burial, wood and bones lose carbon 14 as it changes to nitrogen 14 by beta decay. So here I have shown the carbon 14 goes beta decay and forms once again the nitrogen 14, giving a beta particle. So knowing this, the beta particles are nothing but they are the electrons. So knowing the intensity of this electrons or the electricity, one can find out the, what you can say, the age of the person or the age of the wood. So this is another slide also showing about this carbon 14 dating. So I have explained what I have said. The half-life of carbon 14, you have to remember, that is 5730 years. So this is the what? The living, the C14 to C12 ratio is constant. So you have a line. After death, it goes on reducing. So based on this carbon dating, so let us see some of the problems, how it can be done. Let us take the first one. The wood piece obtained from an archaeological source shows carbon 14 activity, which is 10% of the activity found in green wood. Calculate the age of the wood piece. Here half-life period of carbon 14 is 5770 years. So we know that the equation, the lambda is equal to half-life period. Lambda is equal to 0.693 divided by T half. So 0.693, what is T half? Half-life of the wood? 5770 years. This is equal to the lambda, that is 0.00012 per year. Now I came to know about the decay constant. So what are the given? It says the 10% of the activity. So if N0 is the initial, which we take it as 100, N will be 10 and decay constant is given. Based on this, we can calculate the T with the equation T is equal to 2.303 divided by lambda into log N0 upon N. So that's equal to 2.303 divided by 0.00012 into log the 100 upon 10. 100 upon 10 will be log 10. Log 10 will be 1. So 2.303 divided by 0.00012. So this will be 19191 years. So therefore the age of the wood is 19,191 years. Let us take another example. A sample of carbon from a piece of old wood is found to give 7.6 counts per minute per gram. If freshly cut wood gives a count of 15.8 per counts per minute per gram, calculate how old is wood piece. In this case, what is given? The T half of carbon 14 is given, that is 5770 years. And we know that you have to calculate the decay constant. The decay constant will be 0.693 divided by T half. So it will be 0.00012 per year. So the counts are, the initial count, that is N0 is equal to 15.8 count per minute per gram. N is equal to 7.6 count per minute per gram. And lambda we know. So therefore T is equal to 2.303 divided by lambda into log N0 upon N. Substitute the values, we get 6,099 years. That is the age of the wood is. Let us take the third one. A scrap of paper taken from the Dead Sea Scrolls was found to have C14 to C12 of 0.795 times that found in plants living today. Calculate the age of the scroll. Here also once again the lambda is calculated, that is decay constant, which is 0.00012 per year. Here what is N0? N0 is 1. And what is N? Because it has changed into 0.795. So N is 0.795. Lambda is known. Calculate the T. Substitute the values of lambda, N0 and N. I get 1911 years. This is the age of the scroll. Students, in this class, we have studied the nuclear fusion and various applications of the radioisotopes. And finally, we have done the application of the radioisotopes to find out the age of the wood, that's called the carbon dating, and also the problems on carbon dating. Go through these aspects